which will be moderated by Rita Maliza, SSE MSE PhD. Ladies and gentlemen, welcome to the event Mipa Academic Talk Series 16 Special Lustrum 14 F Mipa with the insightful academic session on lipid production by yeast Rhodotorula strain using biowastes, Tuesday, May 27th, 2025. Ladies and gentlemen, speech by the Dean of Faculty in Mathematics and Natural Sciences, Andalas University. Please welcome Professor Dr. Mike FDMSE.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Good morning, everyone. First of all, let us offer praise and gratitude to the presence of God Almighty because of His mercy and grace. We can gather at this Mipa talk event in the framework of 14 Lustrum at Mipa Andalas University celebration. I would like to extend special greeting to the Honorable Professor Dr. Kazuhito Fujiyama from International Center for Biotechnology, Osaka University, Vice Dean and Manager of the Faculty of Mathematics and Natural Science, Andalas University, Academic Staff, Faculty of Mathematics and Natural Science, Andalas University, and all audience in this Zoom meeting room. It is my great pleasure to welcome each of you to the opening ceremony of Mipa Talk with the topic Lipid Production by Yeast Rhodotorula Strains Using Biowaste. I'm glad to see so many of you here, ready to dive into this important topic. The use of fungi, including species such as Rhodotorula and other oleaginous fungi, to convert organic waste into a source of high-value lipids is a clear example of science-based innovation that is oriented towards sustainability. This technology has great potential to support the bioenergy, bioplastic, and even nutrition industry, while reducing the negative impact of waste on the environment. Even like Mipa Talk play a crucial role as platform for academic, researcher, and student to exchange ideas, engage in critical discussion, and also collaboration. I hope this initiative will inspire the development of outstanding research within FMIPA that contributes meaningful to both society and the environment. I also express my thanks and highest appreciation to the committee that has contributed to holding this event. Finally, I congratulate you on participating in MIPA Talk. I hope this event is useful and provide new insights for all of us. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Thank you so much to Professor Dr. Mike FD for opening remarks. Ladies and gentlemen, moving to the next agenda, we will introduce the moderator who will take the program forward. She is completing bachelor and master degrees at Department of Chemistry, Andalas University, and Doctor of Philosophy PhD in Molecular Endocrinology Division of Histology and Cell Biology, Department of Anatomy, GT Medical University School of Medicine, Shimotsuke, Japan. Lecturer of Biology Department of Faculty of Mathematics and Natural Sciences, Andalas University. Among the achievements she has received include top 5 finalists of L'Oreal UNESCO for Women in Science Indonesia 2023, Best Oral Presenters, National Seminar of Biotechnology, Universitas Gadjah Mada, Yogyakarta 2021, Best Poster Presenter, the 3rd Postgraduate Seminar in Pharmaceutical Science 2020, and the winner of a writing competition, Writing Dikti 2018 Indonesia untuk Citarum Harum 2018. To Mem Rita Maliza, MSE PhD. Time is yours.
Thank you, Intan. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
Assalamualaikum.
Good morning, ladies and gentlemen. I welcome you on behalf of the Dean and the whole committee, especially our distinguished speaker, Professor Kazuhito Fujiyama, to the Mipa Academic Talk Series 16, held with the special 14 Lustrum of the Faculty of Mathematics and Natural Science, Andalas University. It is my pleasure and honor to serve as your moderator for this season. Today's talk series will last 60 minutes and will consist of the 30-minute presentation by our speaker and follow it by a question and answer session lasting 30 minutes. So the topic of today's presentation is lipid production by rhodotorula yeast strains using biowaste. And then I inform you to all the participants, if you have a question during the presentation, you can type your question in the chat box in this Zoom meeting. Before we begin, allow me to briefly introduce our speaker, Professor Kazuhito Fujiyama, PhD. Professor Fujiyama is a professor at the International Center for Biotechnology, IC Biotech, Osaka University. He completed his undergraduate, master, and doctoral studies at Osaka University, specializing in the fermentation technology. And then through his career, Professor Fujiyama has held key academic positions at the Osaka University and has served as a director of IC Biotech since 2017, leading international collaboration, especially with Asian University. His research focused on applied microbiology and glycoengineering. Professor Fujiyama is widely recognized for his significant contribution to biotechnology and also in the international scientific partnership. So at this point, I would like to respectfully invite Professor Kazuhito Fujiyama, PhD, to begin his presentation. Please welcome Professor Fujiyama.
Thank you very much. Can you hear me?
Yes, we can hear you, Professor.
Yes.
Thank you. I would like to show my gratitude to Dr. I'm sorry, I forgot the name of the committee member. Madi? Sorry.
My FD.
Professor
My
FD.
Yeah, my FD. Thank you very much. I'm very happy to be here today, and I would like to introduce my research topics to you. And I guess this is my second time to talk about our research to you. I think I made the first presentation a few years ago, and I appreciate this invitation again by Dr. Rita Mariza. I really appreciate your invitations. And I would like to talk about today lipid production again, but I would like to also share our research progress with you. So may I use my PowerPoint?
Yes, sure, Professor. Please, you can share your PPT presentation.
Okay. Can you see my slides? Okay.
Yes, we can see.
Okay, thank you. So first of all, I would like to, I don't know why celebrate your 70th anniversary International Scientific Forum by Universitas Andalas. And I'm going to talk about these topics today. Wait a moment, I don't know how to pointer show the, yes. So this is our institute, and now I am sitting here, this huge floor. Okay. And today's topics is very helped by these guys, particularly Professor Savitri Dimton in Kasetsat University, Thailand. And today, this is outline of today's talk. So I would like to show some introductions. Then I would like to move on to the Rhodotorula mucilaginosa, Rhodotorula toroides for the lipid production. So what is biodiesels? As you know, one of the alternative and sustainable biofuels. And this is made from renewable, resource triacyl glycerol. And as you know, triacyl glycerol can be obtained from plants, animals, and microbes. But in my today's topics, I would like to show our research on using microbes, particularly oleaginous yeast. So once we get the triacyl glycerol, we can change fatty acid methyl ester by transesterifications. But for this step, we also get plenty of glycerol. So we like to use this waste glycerol for our fermentation experiments. I don't know, I cannot move to the next, yeah. So biodiesel production, as I mentioned, we can use the plants, but I'm very much interested in microbes in our study. And there are some advantage and disadvantage. For instance, plant may need a lot of lands, but oleic acid can be produced a lot using palm oil. The microbes, we do not need a lot of lands, and we can produce the yeast in short times. But as you know, to cultivate the yeast, we need electricity. So we have to consider which is benefit. But beside that, I also would like to show how we can use the microbes oleaginous yeast for the lipid production today. So I'm very much interested in this Rhodotorula mucilaginosa TK16. And this is oleaginous yeast. But this is not Ascomycetes yeast. This is Basidiomycetes yeast. And this yeast, as you can see, orange color. So it means this yeast can also produce carotenoid. Not only fatty acid, but also they can produce carotenoid. So we use this TK16 strains isolated in Thailand. And this is a phylogenetic trees of biotechnological relevant fungi, highlighting the diversity of oleaginous yeast. And you can see blue one, Ascomycetes yeast. For instance, Lipomyces, Clubberomyces, or Yellowia. These are Ascomycetes yeast. Underlying species were described as oleaginous yeast. So, for instance, Saccharomyces cerevisiae, that is not oleaginous yeast. As you know, we can use this yeast for the baker's production or alcohol production. And Lipomyces or Yellowia are oleaginous yeast. And the red one is Basidiomycetes yeast. So today I'm going to talk about this Rhodotorula mucilaginosa, Rhodotorula toroides. Fermentous fungi also shown here, green one. So this is the, for instance, Moquar or Umbellopuscis. These are originous fungi. So today I'm going to talk about Basidiomycetes originus yeast. And here I showed how we can utilize Rhodotorula toluloides for the lipid productions and what kind of research we should do for the lipid productions. So this is the fatty acid synthesis pathway. And in our research, we use adaptive revolutions to get thermotolerant yeast, thermotolerant mutants. Then when we get the mutants, we analyze what kind of genes are upregulated, then what kind of gene may involved in more lipid productions. For the bioprocess optimizations, of course, nitrogen is important, and carbohydrates, crude substrates. Today's topics is related to this one. We use crude glycerol. Trace metal salts, oxygen, temperature, pH, light, electrofermentations. Then our yeast, Rhodotorula yeast, cannot produce this kind of oils yet. So in future, we, if possible, we like to create the Rhodotorula yeast, which can produce these kind of oils. So first, I would like to talk about this Rhodotorula mucilaginosa strains. And as I mentioned, we are very much interested in thermotolerant mutants. I guess I talked about these topics a few years ago, but I like to update some research progress. So Rhodotorula toluloides TK16 can produce more uric acid or A. And uric acid is suitable for the biodiesels. And this TK16 can produce high lipid content more than 20%, rich in uric acid more than 60%. And as I told you, uric acid rich biodiesel can prevent formation of aggregation after a long-term storage. So culture method for the lipid productions, we are interested in this media. So cell growth, YPD media, high nitrogen contents, then composition. So this is the compositions. So this is very typical yeast media, YPD. But for the lipid accumulations, we have to use nitrogen limited media. So CN ratio should be higher. So we can use glucose, but we have to limit the nitrogens. As a carbon source, we like to use the glycerol. Of course, first experiments, we can use glucose, but in future, we like to use the pure glycerol. Then we like to shift crude glycerol. For the industrial scale for the industrial scale fermentations, we have to be concerned about the heat controls. So during the fermentations, yeast sometimes produce a lot of energy as well as heat. So it is very costly to maintain the fermenter at a lower temperature. So that's why we like to obtain thermotolerant mutants. So we study how our originals is Rhodotorula mucilaginosa TK16 can grow at the different temperature. So this is 30 degrees, 32 degrees, 34 degrees, 36, and 38. So our strain can grow well at 30 degrees or 32 degrees. Even this TK16 was isolated in Thailand. So we would like to isolate the thermotolerant mutants which can grow over 36 degrees. So we used adaptive evolutions. So we cultivate our TK16 strains at 30 degree, then shift to 37 degree and 30 degree and 37 degree, 30 degree. We repeated these cycles. Finally, we could isolate some tolerant mutants, just one colony. So one just mutants can grow at 37 degrees. So here is the data. So this is original Rhodotorula mucilaginosa TK16s, and this is the mutants. But TK16 original mutants cannot grow at 37 degree, but our thermotolerant mutants can grow at 37 degree. And interestingly, these mutants can grow at 37 degree, but they can also produce higher amount of lipid. So this is 30 degree and TK16 mutants. At 30 degree, the amount of lipid production should be the same or similar. But at 37 degree, our mutant cells can produce more lipids. So here, a small summary, L11 mutants produce more lipids than TK16, original strain TK16 at 37 degree. So we believe this strain is suitable for industrial applications. Here comes our questions. What factors trigger high temperature tolerance and higher lipid productivity? So we try to identify the gene related thermotolerance and also higher lipid productions. So we do not have genome sequence with TK16 isolated in Thailand. So we analyze the genome sequence, then identify the mutations of the genomes. And here is genome sequence data. Then we found around almost, 20 megabases the genome sequence. Then we also found 19 or 18 contigs. Maybe it means our strain may have 19 or 18 chromosomes. Then we move to the RNA sequence. But before that, we can analyze, or we can get the about 6,800 coding sequence when we compare previously published genome sequence of Rhodotorula mucilaginosa KR. So we use this as a reference strains, and we speculated our strain may have around 6,800 genes. And this is a lipid analysis. So we cultivated our strains like this. So cell growth phase, as I told you, nitrogen rich and also carbohydrate rich media. Then this is nitrogen limited media. Then we isolated lipid and analyze using gas chromatography. And here is the comparison of our mutant strains and the original TK16 strains. And this is TK16s at 30 degree, and this is TK16 37 degree. And this is L11 strain, mutant strain, 30 degree. And this is yellow one is 37 degree. So we can say our L11 strain can grow they can grow very well at higher temperature. And again, we compare lipid productions.
Yeah.
Then we use these strains for the RNA sequence analysis. And here is RNA sequences data. So TK16 30 degree, 37, mutant 30 and 37 degree. So mapping rate around 90%. Then we found very interesting characteristic of the mutants. So L11 at 37 degree, these yellow genes are very much upregulated compared to original strains. So we focus on these genes to analyze which genes could contribute to the thermotolerance and also higher lipid production. Okay, I would like to skip this one. Then we picked up about 42 genes, candidate genes, which may trigger high temperature tolerance and lipid productivity. Then we introduce these 42 genes into the wild type cells one by one. Then we believe this kind of experiments will give us some insights on the mechanisms of enhanced lipid productivity and high temperature tolerance. Okay, then we individually introduce these genes into the wild type cells, then analyze lipid productions. Then here is the data. This is the candidate genes. Then, for instance, for gene 14, 19, 24 may contribute to the higher lipid contents. And again, 14, 19, 24, these three genes may contribute to the lipid amount. So we are interested in these three genes, and also we have more candidate genes in our hands. And here is the data, another data. So gene 11, 14, 20, 25, these, maybe you can see, these kind of 4 genes may change the fatty acid composition. So we are also interested in these 4 genes. Why lipid compositions can be changed by these 4 genes. So this is our short summary, or the summary of the first experiments. So we concluded these genes may contribute to the lipid productions, also lipid compositions change. Then student A work on these 2 genes, and student B work on these 3 genes. Why these genes contribute to the lipid productions or lipid composition change of the lipid compositions. So if I have another chance, I may be able to show why these 5 genes can contribute the lipid production or change of the lipid compositions. So next, I would like to go to this toluene stories. So mostly, I would like to talk about the lipid production by these strains using bio-waste. So lipid toluene, again, they can accumulate lipid more than 70%, up to 70% of its cell dry weight. Then we can use these strains to produce fatty acid using bio-waste. So in this study, as I told you, we are interested in the use of crude glycerol, which can produce by fatty acid production from oil. So this is a newspaper of the Malaysia. So they used cooking oil. They are interested in used cooking oil recycling mandates. So maybe in even Indonesia, you use a lot of cooking oil for your cooking, right? So this is the waste oil. So we are interested in this cooking oil to produce fatty acid. So before study, as I would like to explain again, we use the cell growth study and cell growth phase, and this is the lipid accumulation phase. But cell growth phase, YPD, and lipid accumulation phase is glucose. But instead of glucose, we like to use the crude glycerol. To use the crude glycerol, we have to overcome some issues because this is a waste, okay, during the production or purification of the fatty acid. So they have some impurities. So here is the cell growth when we use pure glycerol and crude glycerol. Crude glycerol cannot grow. When we use crude glycerol, the yeast cannot grow well. So we consider this growth is inhibited by something. So we study what kind of impurities in crude glycerol. So maybe methanol, fatty acid methyl ester, fatty acid soap, and salt. So proper treatment for neutralization is desirable. Then we can use this crude glycerol for the fermentations. So in this study, we use Dorotorula toluloides NBRC 0880. So this is the type strains, and we got these strains from the Japanese cell bank. Then we use these strains, not from the Thailand. Then we did 3 experiments: characterization of crude glycerol and the effect of impurities in crude glycerol on yeast cell growth, and neutralization of crude glycerol and its utilizations. So this is a crude glycerol. In this study, we purchased this crude glycerol from these companies. I don't know in Indonesia, but in Japan now, this kind of crude glycerol is very attractive source for the production of airplane diesels or airplane oils. So this is the price is getting higher. The physical characterization of this crude glycerol is dark brown and pH around alkaline conditions. Then chemical composition. So this crude glycerol, about 40% is glycerol, and methanol is 11%, fatty acid 8%, fatty acid methyl ester is 6%, fatty acid soap is 30%, and salt, for instance, potassium and sodiums. So we are suspicious with this fatty acid soap as the inhibitor. The effect of impurities on the cell growth was examined. So we changed methanol, uric acid, methyl urates, and potassium chlorides, but those kind of impurities may not significantly affect the cell growth. Potassium chloride is a little bit effective, but not so significant effect. But potassium orotate surely inhibit cell growth. So we believe this potassium orotate is a key inhibitor of the yeast cell growth. So we tried the neutralizing process. So we changed the pH of this glycerol to pH 6 using hydrochlorides. So you can see two phases, organic layer and water layer. Organic layer may contain fatty acid, methyl ester, and fatty acid. Water layer may contain glycerol, methanol, and potassium chlorides. So through this process, this potassium orotate may be excluded. So we can use this neutralized crude glycerol. I mean, this water layer can be used as crude glycerol or neutralized crude glycerol for the fermentations experiments. So neutralizing crude glycerol as a carbon source, we grow our cells using this neutralizing crude glycerol, not using glucose. Lipid production phase, as I mentioned before, we have to use nitrogen-limited media, and we also use this neutralized crude glycerol. Then we tested the lipid production. So here is the data, and the growth curve of the rhodotorula cultured by different carbon source. So black one is glucose, and yellow one is pure glycerol. Then this is crude glycerol, and this is neutralizing crude glycerol. So as you can see, crude glycerol, the yeast cannot grow because of sodium orotate. And pure glycerol, the growth is good, but compared to glucose, a little bit slow. But when we use neutralizing crude glycerol, shows very similar growth as the pure glycerol. So we consider neutralized crude glycerol can apply for the cell growth of rhodotorula toroides. Then this is the stain of the cells during the cultivations. So lipid production by rhodotorula cultured by different carbon source. So you can see this is glucose, and this is pure glycerol. This is neutralized crude glycerol, and this is crude glycerol. And we stain using Nile red. So this pink one is a lipid droplet, and a blue one is cell walls. So you can see a lot of lipid droplets when we cultivate with glucose or pure glycerol or neutralized crude glycerol. On the other hand, when we use crude glycerol, we cannot see lipid droplet here. So this data shows crude glycerol cause cell defect, or they can uptake the lipid production, but they cannot produce the lipid. So from this data also shows neutralized crude glycerol can be applicable for the lipid productions. So here is the data, lipid production of rhodotorola toluloides cultured by different carbon source. So again, this is glucose, and this is pure glycerol. This is neutralized crude glycerol. And this one, crude glycerol. So crude glycerol, as you know, the cells cannot grow. That's why they cannot produce the lipid. Then this is glucose. Then they can produce a lipid. And pure glycerol or neutralized crude glycerol can also produce a lipid. But compared to the glucose, the production of lipid is a little bit low. Here is the data productivity. Of course, glucose is much better, but the neutralized crude glycerol is comparable to the pure glycerol. So pure glycerol or nitrogen neutralized crude glycerol, less productivity, but similar lipid yield. So we believe we can use neutralized cell growth for the lipid production. Okay, so we try this neutralized crude glycerol for the 5-liter fermenter. Then we divided into the 3 phases. Of course, this is the 5-liter fermenter, so we cannot change the culture media drastically. In a small scale, we can cultivate the growth phase in 25-liter flask. Then we can harvest the cells and transfer to the nitrogen-emitted media. But when we use this kind of fermenter, we cannot change or we cannot harvest the cells. So for phase 1, this is the batch method. Then we use the carbon source, glucose. Then 2.50 liters. Then the second phase, we use fed batch culture systems. Then we control dissolved oxygen. Then we use the glucose. So the cells start growing. Then this fed batch culture cell can grow very rapidly. Then the phase 3, we use neutralized crude glycerol for the lipid production. Then we feed this neutralized crude glycerol constantly. So finally, 4.5 liter, we cultivate like this, then we accumulate the lipid. Oh, sorry. Then this is the data. So, sorry, maybe something wrong. Sorry, this is the data. So I cannot show this, sorry, today lipid productions, but I like to show how we cultivate our cells.
Compression molding atau injection molding. Ini agak
Today, I would like to show how we cultivate, how we use the neutralized crude glycerol for the large-scale productions. I mean, even large-scale production, but we just use the 5-liter fermenter. But in future, I would like to use this system for the large-scale fermentations. So I would like to finish my talk. So thank you very much for your kind attentions. So in future, again, we like to use the crude glycerol, but to use the crude glycerol, we have to neutralize. Then we can use the neutralized crude glycerol for the fermentation to get more fatty acids or more lipids. Okay, so thank you very much for your kind attentions, and I hope I could have some questions. Thank you.
Thank you very much, Professor Fujiyama, for your insightful and inspiring presentation. I hope all the participants enjoy it and then benefited from these amazing sessions. So today we learned about the potential of Rhodotorula yeast strains, produce lipid from biowaste, and then how to process the optimization strategy. This yeast can produce lipid in the, and then the implications for sustainable biotechnology. So with this insight, let us now move to the discussion session. To all participants, please feel free to submit your question, and maybe you can raise your hands or ask directly to Professor Fujiyama, or type your question in the chat box in this Zoom meeting. And then maybe you and also mention your name and also institution. So still waiting for the question. Okay. We got one question from Diana Vandawelia. Dear Professor, as far as I know, catalysts are also used to produce fatty acid. Have you ever compared which is more efficient using yeast or catalyst? From Dr. Diana, Professor.
Okay, thank you very much, Professor Diana. In our case, we use the crude glycerol as a carbon source for the yeast fermentation. So yeast eat the crude glycerol, then they can produce triacyl glycerol. So that's why a little bit different from the chemical catalyst. So we give the feed to the yeast, and yeast produce triacyl glycerol. Then we can get the fatty acid from the yeast. So the story is a little bit different. Is it correct? My understanding is correct, Professor Diana?
Actually, I'm also not too sure. I just want maybe the insight from you, so I also can improve my knowledge maybe. So it's very thank you for your explanation.
Yeah, so Professor Diana, if possible, I'd like to have your idea how we can apply catalyst. Sorry, I am more biology people, so
Yes.
I'm happy to have a chemical insight from the chemist.
Yeah, actually, I think this is also our colleague, Dr. Shukri's research topic. Dr. Shukri is here, right? Maybe Dr. Shukri can explain more detail and clear regarding to this. I cannot see him here. Just it, Prof. I think I also not. This is not my expertise. As far as I read and I know about my question in the chat box.
Okay.
Yeah,
Thank
sorry,
you.
Prof. Yes. Okay.
Thank you.
Thank you.
Okay. Thank you to Dr. Diana. So, okay, we're waiting for the second question from all of the participants. During the waiting the question, maybe I wanna ask something. We have the question. Maybe I read the second question from Ibu Amelia. Dear Professor, I'm Amelia from the University, Bung Hatta University, Universitas Bung Hatta. The topic was very interesting. I want to know what specific characteristic of microorganism that can we use for this fermentation process.
Okay, so thank you very much, Dr. Amelia. In our yeast, which can produce more lipids. That's why we call it oleaginous yeast. And Saccharomyces cerevisiae, like a baker's yeast, they cannot produce a lot of lipids in the cells. So we have to screen the oleaginous yeast for the lipid productions. Then some oleaginous yeast may have very specific potentials for the lipid production as well as accumulation of the lipid. So we have to use the oleaginous yeast for the lipid productions, but for the engineering of the yeast cells, Saccharomyces cerevisiae is very promising the host. So you can introduce a lot of genes into the Saccharomyces cerevisiae. And also you can engineer chromosome of this yeast very easily compared to Rhodotorula toroides. So depending on your research or depending on your interest, you can choose what genes you should work on. So is it okay for my answer?
So, Dr. Amelia, is it okay? Thank you, Prof. Okay.
Thank you.
So, all the participants maybe have questions for Professor Fujiyama.
So
Okay. We have another questions. Professor, I can read the question. My name is Enda. So Enda is the biology student from Department of Biology. So she asked you, why did this research choose the glycerol and CG, crude glycerol, and then the other one NCG to optimize lipid production from strains instead of using other nutrients? Thank you, Professor.
okay, thank you very much. We are interested in bio waste. So as I mentioned, crude glycerol is produced from cooking oil. So I don't know in Indonesia, in Japan, we try to use the waste cooking oil for the production of oil, or I mean airplane oil or car oil or biodiesels. So once they harvest the cooking oil, the company try to extract the fatty acid from triglycerol. Okay, then when they extract the fatty acid from triglycerol, glycerol is produced as a byproduct. So mostly they throw away or they try to use this waste glycerol as the feed, like a cow or a chicken, or sometimes they spread into the vegetable farm. So they just throw away. So I like to use this waste glycerol for the another production of fatty acid. That's why we try to give this crude glycerol to the yeast. So yeast can eat, then produce another round of triglycerol. So that's why we use this crude glycerol as a carbon source for the fermentation of yeast. Is this okay to?
Yeah, very clearly answered, Professor.
Yeah.
Okay. Now we continue to the next question from Ibu Diah Fitriani. So using this yeast and glycerol as the carbon source also produce beta carotene? Did you also extract the beta carotene? I would also like to ask about the lipid extraction process.
Okay, thank you for your questions. Yes, this yeast strains can produce the carotene, beta carotene. That's why you can see orange color or sometimes red color. Now, during the extractions, of course, we can extract the carotene, but we haven't analyzed carotene yet. But one of my students, PhD students, is now setting up how to analyze carotene contents. Yeah, so he just made a publish his paper when we change the iron, Fe iron, not iron, Fe contents, then the cells can accumulate more carotenes. So he is interested in how the concentration of iron change the carotene content. So Daya, thank you very much. Maybe you are from Surabaya?
Yeah, they are from ITS, Institute Technology Surabaya. Is it correct?
10 November.
10 November.
Yes.
10 November.
Yes, I'm from Surabaya.
Okay, from Surabaya. So she also asking about the lipid extraction process, Prof. So
Oh, lipid extraction process. We just use the organic solvents. I'm sorry, I don't remember details. If you need, I would like to give you through Dr. Rita. I'm sorry, I don't know your email, so I will send our protocol to Rita Mariza, Dr. Rita Mariza.
okay, Prof. So maybe Ibu Dia can send your email in this chat box. Maybe later I will send you your the protocol. Okay. Yeah, I have also sent the question with Ibu Dia, Prof, about the Rhodotorula mutant and then wild type. Yeah. So there's beta carotene, fatty acid, so many, maybe another metabolic secondary, we can find different amount later after optimization the temperature. So in this research, beta carotene is important for the vitamin A. So that the professor PhD student is already reported and published the journal. So another step could we make control to another gene expression also up and down regulated for this expression?
Okay, thank you very much for your questions. We have not studied the gene regulation yet under the starvation of iron or iron-rich media. So we like to study how iron concentrations can change the gene expression. The gene should be related to the beta carotene for the production. Yeah, that is very good, interesting questions and suggestions. Thank you very much.
You're welcome, Professor. So our participant, you can raise your hand directly, ask directly to Professor Fujiyama
I like to add one more issues. So as
Yes.
I mentioned, our yeast cells cannot grow well in the presence of crude glycerol. So to facilitate use of crude glycerol, one Indonesian student, he is from UGM, now trying to get the mutants which can grow in a crude glycerol. So I hope he will show how we can produce the fatty acid using his mutants, using crude glycerol as a carbon source in future. Thank you very much.
Yeah, interesting, Professor. So, your presentation informed that you got the strain from the Thailand, right? Rhodotorula. Uh, how about the Indonesia? Have you tried to explore the Rhodotorula strain in the Indonesia by ways? Okay.
Okay, thank you very much for your questions. I also would like to use the bio waste from palm oil production. So if possible, I would like to isolate the Rhodotorula strains from Indonesia, and if we could have any collaboration with Indonesian professors. But now we have, I mean, Japanese has a problem with Indonesian government policy because your government policy is very strict in terms of biodiversity.
Yeah.
So we need some collaborator in Indonesia. Then we can do some research in Indonesia. If not, it is very difficult to get the strains from Indonesia now.
Okay, Professor. Maybe we make some like MOU first, and then
Yeah,
we also have like grant for research collaboration, like that. Okay,
yeah. Then we can do some experiments with you.
Yeah, Professor. Thank you very much for the change, Professor. Now we have another question from Samudra Ilham. Samudra Ilham is a biology department student. So good morning and thank you so much to Professor Fujiyama for the presentation. Insightful research. My name is Samudra Ilham. So undergraduate students from the biology department. I have an interest in synthetic biology. I would like to ask a question related to your future plan for Rhodotorula mucilaginosa. Why is it necessary to reintroduce right, for a promising mutated gene into the wild type strain instead of simple propagating the selected mutant or repeating the adaptive evolution experiment?
Okay, thank you very much for your questions. Yes, of course, we can use the another adaptive evolution experiments. Yes, that is true. But Samudra, please understand, to get this L11 mutants, one PhD student spent his 9 months. He spent 9 months to isolate the mutants. Okay, so it is a bit difficult to repeat this adaptive evolution experiment because the strain is already, how to say, grow up to 37 degree. So from now, for instance, 42 degree or 40 degree, it is harder to get the mutants. That's why we stop this kind of experiments. Then our interest is shifted to why these mutants can produce the more repeat. So what enzymes is more produced, or what enzymes can involved in the more repeat production? So to understand that, how to say, introduction of the upregulated gene into the wild type cells, that is easy study or easy protocol. That's why we chose the upregulated gene, and we chose upregulated genes, and we introduced those genes one by one into the wild type cells. Then we can reproduce the mutant cell, transformants, which can produce more lipid compared to the wild type. So we try to accumulate very basic informations. Now you understand?
Okay, Ilham.
Thank you for your answer, Professor Fujiyama. Thank you very much.
Thank you.
So thank you to Ilham for your question. Professor, is it possible to share your PPT presentation for us?
Yeah, okay.
Yeah, okay. Later I will contact by email, Professor. Okay. Thank you to Professor Fujiyama. There are no future question, I think. We have now reached the end of these sessions. So on behalf of the Dean and Committee, I would like to express our sincere gratitude to Professor Fujiyama and to all the participants for your active engagement and attentions. I will now return the session to our Master of Ceremony, Ms. Intan. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Ladies and gentlemen, we thank you very much to Professor Dr. Kazuhito Fujiyama, International Center for Biotechnology, IC Biotech University, Osaka, Japan, for insightful academic session on lipid production by yeast, Rhodotorula strain using biowastes, and also our moderator, Mem Rita Maliza, MSc PhD. Ladies and gentlemen, awarding certificate to the speaker and moderator by the Vice Dean of Faculty of Mathematics and Natural Sciences. Please welcome to Professor Dr. Admin Nazra.
Okay, thank you. The Honorable Professor Fujiyama, on behalf of the Faculty of Mathematics and Natural Sciences, Andhras University, it is our great honor to present this certificate of appreciation as a sincere token of our gratitude for your outstanding presentation and valuable contribution to today's webinar. And we deeply appreciate the time and expertise you have generously shared with us, which has truly enriched our academic discourse and inspired further collaboration. Thank you once again. See you at others webinar. Thank you.
Thank you. Thank you very much for this certificate. And I'd like to add one more issues. So if possible, I would like to see you in Osaka, the University of Osaka.
Okay, thank you.
From April, the name of our university has been changed from Osaka University to the University of Osaka.
Oh, I see. Okay.
Yeah, I don't know why, but our president changed the name.
Yes, thank you.
Thank you.
Okay, thank you. Okay.
Intan, ada Intan.
Ladies and gentlemen, moving to the next event, the awarding prize to selected audiences. Congratulations to Amelia from Bung Hatta University, Enda Tarni Biology University Andalas, Dia Fitriani dari ITS. We kindly ask the door prize recipients to contact the committee after the event. or by email by secretariatfmipaua2@gmail.com that we adding in chat Zoom column. Ladies and gentlemen, before we close our talks, let's take documentation section. Please open up your camera.
Okay, everyone, I take a picture from this Zoom. Please turn on your camera. And I take slide 1. Please say cheese. 1, 2, 3. Okay, and slide 2. 1, 2, 3. Okay, thank you all.
So thank you very much. And if you could have a chance to visit Osaka Expo 2025, please also stop by at Osaka, the University of Osaka. We very much appreciate your visit at our university anytime.
Thank you very much, Prof.
Thank you very much, Professor.
Ladies and gentlemen, finally, this is the end of Mipa Academic Talk Series 16 with the topic Lipid Production by yeast rhodotulustran using bioestes. As Master of Ceremony, Intania Azara Putri Sanes, we say thank you so much for your attention. Have a good day and wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Professor, once again. Thank you.
Thank you.
Thank you so much, Professor.
Thank you. Take care.
Yes,